with a state-of-art laboratory and operational across the United States, the United Kingdom, Germany, China, Singapore, Japan. Novogene has established a strong global presence to support scientific and clinical advances worldwide with a highly skilled and interdisciplinary team. Novogene service covered the full spectrum of multi-omics from genomic, transcriptomics, proteomics, and metabolomics services, empowering researchers and organizations with cutting-edge multi-omic solutions, supported by our high-throughput sequencing platform, automation workflow, and comprehensive bioinformatic analysis. Through extensive collaboration with leading academic institutes, hospitals, pharmaceutical companies, and agriculture enterprises, Novogene continues to share the future of multi-omics research and precision medicine as your trusted partner. And it's our great honor and privilege to have Dr. André Uitterlinden here to get inspired by the Genome of Europe project, coordinated by his leadership and visions in the field of human genomics. Dr. Uitterlinden is the professor of complex genetics and also population genomics. He's also the head of the genetic laboratory at Erasmus Medical Centre in Rotterdam, the Netherlands. Welcome, Professor André Uitterlinden. Professor Uitterlinden research involves identifying genetic factors for common traits and disease to apply the discoveries for clinical practice and the treatments. He has been also focused on working on the 1+ Million Genome project in Europe and implementing genetic information in healthcare and prevention settings. In 2025, he initiated the Human Genomic Facility, which is now one of Europe's largest genomic facilities, providing services for DNA isolation, genotyping, sequencing, and data analysis, and handling millions of samples globally. Between 2005 and 2010, he initiated the widely used array GWAS and DNA RNA sequencing database for two well-known Erasmus cohort studies, namely the Rotterdam study and the Generation R birth cohort, with a large group of population of participants. In addition, he is one of the founders of several global GWAS consortiums, and he has co-authored over more than 1,500 papers with a high index more than 190s. In 2020, he became members of the coordination team of the European 1+ Million Genomes Initiative and has been leading the Genome of Europe project to establish more than 100,000 reference genomes. The Genome of Europe project brings together more than 30 European countries, 41 institutes, and more than 200 top scientists through a collaborative effort to sequence the first 100,000 genomes, establishing a concrete foundations of European reference database for the development of personalized medicine and health approaches. The data will be accessible via the Genomic Data Infrastructure. In today's webinar, Professor Uitterlinden will be sharing the latest progress of Genome of Europe and highlight how large-scale sequencing is reshaping and pioneering research and healthcare. He will also explore the potential of genetic data in disease prevention and share his highly valuable visions in the future of genomics. Besides his numerous scientific contributions, he has been dedicating his endeavors in education as well, supervising more than 40 PhD students and is leading annual courses on complex genetics and genomics. Professor André Uitterlinden, please, the stage and the microphone are yours.
Well, thank you very much, Wijn-Lin. Too much honor. I will start sharing my screen. I think you have a visual now.
Yes. At least I can see your deck. Yeah.
Okay, complete screen. Well, thank you very much, and really, it's a pleasure and an honor to be speaking on this fantastic forum you organized. So, as you introduced already, what I will do, we'll of course speak about the Genome of Europe, but also give you a brief introduction of where my research is coming from and where the 1 Million Genomes project is coming from and why we are doing that, in fact. And these opening slides must be recognizable by all of you. This is how we do research in general. Research with incomplete means. Of course, they are improving all the times. And then we find something, but then we have to start deciphering it. And you see here a well-known example, the disc of Vestos, found on the beautiful island of Crete in near Greece several thousand years ago. And we still have no idea what it means because we lack a reference. Like the Stone of Rosetta provides a reference for 3 languages, this one is still undeciphered. And that's a very usual occasion happening in science as well. We need reference genomes, and that's why I will speak about the reference genome of Europe. And of course, once we have done all that fantastic research, we start implementing, but of course, we always have to be aware that we have to do this for the best of humankind. So we have to follow the oath of Hippocrates. But we should also be aware of, let's say, unwanted side effects that might occur because of this. So there might be boxes of Pandora to be opened, and we have to be very careful of this. I have no disclosures, actually. Sometimes I get paid to, but not this time, to speak for companies and advise them a little bit. But in general, my research is funded by public resources. I receive only one salary. I just retired also as a disclosure. And I do not hold any stock in any company. This is, of course, the well-known starting point of, I think, all of the genetic endeavors we do. The human diversity, which is bewildering, as you can see here. Some well-known examples can be spotted. And of course, the interest we have here working in an academic hospital is not so much the behavior or how they look like, but the diseases that people develop. And if we understand that better, maybe we can intervene in that process and also help treating them in a more optimal way. And that's why DNA research has been so important, because it has been very valuable in unraveling, let's say, the architecture of diseases in general and understanding much more about the causes of diseases, which, of course, DNA is a blueprint of life, should be found somewhere down the line in the DNA code. So to summarize, a lot of research over the past decades is that we learned that variation in the DNA sequence between human individuals is involved in explaining parts of the differences between humans in, for example, development, in how babies are born and how they develop, in how people look like, tall people, small people, fat people, thin people, etc., color of your eyes. The behavior, you just saw some fine examples of what might be termed aberrant behavior. Also, that is under partly genetic control. Aging, which is sort of a dustbin of all the misery that's happening once we progress in time and reach old age. And of course, here in the hospital, there's a big focus on particular diseases, and not only in diagnosing disease, but also treating those diseases, and maybe in a more public health setting, try to prevent diseases, for example, by predicting risk. And I'll come back to that later. Now, all of this together had led to this new concept, although it's not new. If I speak to clinicians, they say we've been doing personalized medicine for 2000 years already. That's not new. Precision medicine might be a better term, because now we have a lot of molecular data which we can use for all these different applications of clinical science. So that's where the molecular science and the molecular genetics comes in. Now, why are we so interested in DNA variation? As I said already, DNA is a blueprint of life, so it's encoding how life works in general. And what I wrote down here is important not only for the rare diseases, the Mendelian mutation, but also for common variants, which we learned loads of them through genome-wide association studies, GWAS. Knowing the correlation between a DNA variation and disease or a phenotype later on helps us to understand the biology. And that holds true for the mechanism. So we know how DNA is encoding a protein, that protein influences cellular processes. And that helps us to develop new treatments, because once we understand the mechanism, we can identify, let's say, interventions in that. That could be drugs, but can also be lifestyle or other types of environmental exposures we can monitor and change. What is also important is that DNA is a stable molecule, so you're born with it and you die with it. So that allows you to predict, once you know that correlation between a variation and a endpoint, you can determine that variation somewhere earlier in life, maybe at birth, but we can discuss about that. and then predict what a risk a certain person has based on this set of DNA variations. One particular application, but I will not discuss that today, is called Mendelian randomization with polygenic risk scores. And that has been an important result of all the GWASs being done over the past decades. And that has helped a lot to understand much more about the causality of certain factors that we are already measuring in people, like serum CRP levels or cholesterol levels, etc. Are they causal or are they bystander effects? And that has been important in also treating people for these biochemical markers. Maybe more important, I'll come back to that, is the early diagnostics we can do based on the DNA information which we determine through the different technologies to predict risk of disease. And that is, of course, is important for prevention, but also progression of disease. And that's a clinical interest. Once you have a disease, how do you progress? Are you a fast progressor or a slow one? Is it leading to, let's say, very detrimental endpoints, or is it a slow, mild phenotype of the disease? And that, of course, can be important to direct interventions in there based on that clinical knowledge. And last but certainly not least is the realm of pharmacogenetics, where we have loads of drugs already all there on the market. And as we all know, they don't work in everybody. And sometimes this is an abysmal small number of patients that actually profit from a certain drug. And we've learned that several genetic markers might help us to understand who will benefit from a drug and who will not. And that holds in terms of adjusting the dose, but also in predicting side effects of drugs. Now, an important question is, of course, for all these applications, how much variation is there in the genome? And I would like to stress that you see that on the top left corner here, that's based on a lot of these big famous projects like the Human Genome Project or the 1000 Genome Project or GNOMED or DB-SNP is collecting all this information of all these fantastic projects, TopMed, for example, most recently. And that has led, and that you see that on the right lower corner, has led to an enormous growth in the knowledge of variations. And the current estimate is that over 1 billion nucleotides in the DNA molecule can vary between human individuals. So that's 1/3 of your DNA that can vary between you and your neighbor. Of course, the vast majority of that will be very rare, but a substantial proportion, somewhere between 10 and 20%, we guess now, is quite common actually. So in other words, there's an enormous variety of DNA sequences between human individuals. And we've only scratched the surface by all these sequencing efforts, because if you add them all up, only about 6 million people on the planet have currently been sequenced. And that's, of course, a really tiny fraction of people. And already that has given this insight that there's an enormous amount of DNA variation. Now, based on that knowledge of DNA variation, all the different technology, now I'll not go into detail today about that, that has been of big help in uncovering all these variations that matter for diseases. And you see here that is mainly a function of the frequency of that variant and the effect size they have. If you look at the top left corner here, you see the situation for the very rare diseases, the typical Mendelian clinical genetic diseases. They are caused by very rare variants with a very big detrimental effect, usually very early on in life, quite visible in people carrying these mutations. And once you have that, let's say, that pedigree collected together with enough meiosis in it, it now takes a matter of weeks to uncover the causal DNA variation for that particular Mendelian disease. There are about, 8,000 of those Mendelian diseases. Over half have been solved because of this approach, but there's still, of course, many to tackle with that through the DNA sequencing. And what we've learned is that many of these variations are also clinically actionable, meaning they can be used for not only diagnosing patients carrying those diseases, but also help in prediction of risk, for example, for couples that have a child with. So clinically very important also in the realm of reproduction. And now there, of course, as you know, there are some projects looking at newborn screening, for example. Can we use this information to very early on in life screen for these Mendelian mutations? Very interesting topic. I will not go into that today. On the right hand, you see the other, let's say, part of the spectrum. Those are the common diseases. And those are, in fact, the big clinical burden at the moment, globally speaking. These are the big diseases of the aging populations: Alzheimer, diabetes, osteoporosis, cancer, you name them. We are all very familiar with it, as are the clinicians here in the hospital. Those are the patients they see every day in the dozens. So that's where they want to get new grip on understanding the causes of those diseases and come up with maybe new interventions like drugs or lifestyle interventions. And what we've learned through all those genome-wide association studies by arrays, by genotyping, very large collections of biobanks and cohort studies, is that there are not one variant per disease, but thousands of these variants per disease. So that, of course, has led to some confusion in the beginning because the effect size of individual variants is small. But collectively, we can now bundle them in so-called polygenetic risk scores. They have effect sizes which are equal to those of the single Mendelian mutations. So clinically speaking, those PRSs are equally important as those Mendelian mutations, but then valid for a much bigger proportion of the population because they are so common in the population. And I'll come back to that later. And that has led to this, let's say, depictions of the architecture of disease, the genetic architecture of disease. If you follow a particular trait, and you see that on the left, on the y-axis, take, for example, cholesterol level, that follows a what we call Gaussian distribution in the population. So you have very few people with very extreme values, and the bunch of most people will carry a, let's say, normal level of cholesterol. And we've learned now that those Mendelian mutations explain most of the extreme cases of these values, in this case cholesterol, but you can also look at glucose or bone density for that matter. Whereas the vast majority of the people in the population are explained by these polygenic risk scores carrying all these SNPs we identified through the genome-wide association studies. And below on the graph, you see the techniques that have helped us to uncover all these. So that has also led to a different view on genetics, also of Mendelian genetics, but also of common genetics. We should take a much more holistic view of the genome. And that involves not only looking at rare variants, but also at common variants. So it's the combination of common and rare variants that explain the genetic architecture of basically every disease. Now, very important in this, and that's why I'm happy to speak on this forum, is technology, because that really has been pushing our field forward, not only with sequencing, of course, I will come back to that later, but also in this case by arrays, which is a tool which is, in my view, a little bit underappreciated, because it has helped tremendously to generate all those data for the genome-wide association studies, and many more people are being genotyped by arrays than people are sequenced. As you see here, an important part, of course, of all the negotiations we do with companies like Novogene, but also the big providers like Illumina and PacBio, etc., is the price of this determination. And you see here the development of arrays that was initially quite expensive, almost 1,000 euros per sample to be done when I started to run the genome-wide association studies. And because of negotiations with the companies, Affymetrix and Illumina, me, but also with some colleagues, of course, we talked that down to what it is now, less than 30 euros. And if you do it even large scale, it can get close to 10 euros per sample. And that allows you to genotype at least 1 million DNA variations, we know upfront. So it's a very good technology for screening in large scale applications. Sequencing, of course, is the technology to uncover and to discover these variants. But once you know them and you want to screen them in large data sets, array is really a preferred platform for that. Not only are they so much cheaper, but they're also customizable, meaning you can only put variants on it which you want to see. So these can be clinically actionable variants, and you will not be burdened by what is called the variants of unknown significance, the VUSs, which is a headache, of course, for all the clinical genetics doing whole genome sequencing. The throughput is extremely high, and that's why they are so very useful for the large-scale screening operation. And that also comes with a very easy IT footprint and data handling, which allows them to be interpreted in a quite straightforward manner. So that has led to some programs, and I'll come back to that later, where these tools are used to use genetic information in clinical settings, but maybe also in public health settings. But of course, in order to do that, we need good reference data. And that, I'll come back to that in a moment, has led to this Genome of Europe project. A final word about the arrays, because there was initially some discussion about them that, are they useful to type very rare variants? And that's why we and some other people also have done some work on that. And the answer is yes, they can. And you see here a few publications and some others which are still in the making, where we show this effect for different disease spectra where Mendelian mutations are underlying this. For example, for familial hypercholesterolemia, we showed in a paper that it is possible to genotype thousands of mutations found in FH with an array in a large-scale screening setting. So that is now sort of revived the interest in these arrays, also for screening for Mendelian mutations in different kinds of settings. And again, the price is, of course, a quite important determinant here. Now, why is there so much enthusiasm about polygenic risk scores? Well, that also is reflected in this slide. Because they are growing and growing still because of these bigger and bigger sample sizes we have, explaining more and more of the genetic architecture of all these diseases. So you see here in 2010, when we were doing a few of these GWASs already, we had quite big sample sizes for that time, running into the 100,000s, but explaining about a few percent of most of these diseases. So enthusiasm was low at that time, but we saw progression coming on. And if we look now, 2022, a few years ago, I should update again this in a few years' time. We see, for example, for height, we have now genotyped over 5 million people, identified 9,000 SNPs explaining the height genetics. So that shows how the progress in sample size has really determined our understanding of the genetic architecture of height in this case. And you see the same thing happening for all these other diseases and phenotypes. So many of them are now also underway in genotyping millions of people and understanding much bigger proportions of the genetic architecture, leading to much more proficient polygenic risk scores. A few words about the polygenic risk scores. So what is that? So it is a score, so everybody has a value based on the composition of the alleles in such a polygenic risk score. And you can be somewhere in the middle, let's say the green cloud of dots here, meaning you have a normal polygenic risk score like most people in the population, but you can also be on the left side where you are sort of protected for this particular disease risk based on your polygenic risk score, having a very low risk for that. But of course also you can be on the right side where you have an extremely high risk for that particular disease. And again, if you're in the top 1 to 5% or so, depending on the disease, that risk is equal to that of a single Mendelian mutation, which we all see as a very clinically actionable variant at the moment. And that also holds true for these top percentiles of the polygenic risk score. On the top, you see a few of those diseases where people have looked at and say, well, we have now good proficient polygenic risk scores, which allow us to predict this risk in a robust and useful way also in clinical, but maybe more importantly also in public health settings. And that's shown in this figure below the graph here. You see this people being genotyped for that, leading to different types of preventive measures you can take. So if you're in the middle distribution, you can undergo, let's say, the normal routine screening. If you're at the lower end of it, maybe that screening is of no added value to you because your standard risk is already so low based on your genetic composition. Whereas if in the high end of the risk distribution, you should undergo this screening more frequently or at an earlier age, and I'll mention one example a little bit later. And that has led to this enthusiasm about polygenic risk scores, not only in clinical diagnostic settings, but also in public health screening settings, where we have several of these screening programs ongoing now. For example, for the big cancers like breast cancer and colon cancer, at least here in the Netherlands. An example that we are following here at Erasmus Medical Centre is how can we use all this genetic information that we have generated, globally speaking, for not only the Mendelian genetics, but also the polygenic risk scores in a setting where clinicians can actually use this in the clinic. And that's the idea here of the so-called GOAL project, genotyping on all patients. I'm a big football fan, so that's why I like the term GOAL here. And you see here on this little screen that the clinician and the patient will discuss briefly this list of genetic variants that have been genotyped before the patient has entered the hospital. So the clinician sees then a big screen of green, meaning nothing to do, nothing to worry about, a few reds where they say, hey, stop, there's a clinical actionable variant here which you can act upon actually. And that can be, for example, the discovery of an FH mutation, which this person was not aware of. And he came in with a very different complaint for that. And the good news is that mutation can be treated very well with providing statins to this patient. And of course, we are now discussing this with several clinical groups. You see them listed here, not only in the cancer field, but also in the ophthalmology, eye diseases, cardiovascular people talking about familial hypercholesterolemia and coronary artery disease, pharmacogenetic information, of course, and also important, that used to be my hobby in the past, the locomotor diseases, osteoarthritis and osteoporosis, where also very good polygenic risk scores exist now. So with those clinical departments, we're setting up several pilot projects in Erasmus MC, together with our, as Wylin mentioned already, our fantastic core facility led by Joyce van Meurs, where we provide this information based on the area genotyping to different clinical departments, like he mentioned, for copy number variants for the clinical genetics department, but also methylation profiles for not only clinical genetics here in the Netherlands, but also the Department of Pathology for profiling tumors, which have a very immediate effect on what and how to operate. And of course, we're also discussing how can we use these polygenic risk scores, which are a little bit more experimental, for example, for improving breast cancer genetics, but also use them in predicting eye diseases, for example, macular degeneration as one of the best polygenic risk scores. And people here at the ophthalmology department use it now to predict the risk for that particular disease in their patient population. As you might know, there are also several clinical trials going on now, because in order to convince clinicians, you need clinical trials at the moment. That situation might change in the future, but we'll have to see. But there are several clinical trials ongoing now. Is, for example, polygenic risk score a useful tool in predicting risk for breast cancer? As you know, there are several breast cancer screening programs based on mammography. And that's where a lot of enthusiasm is now to use those polygenic risk scores for predicting risk of breast cancer in a much earlier phase. So run the polygenic risk scores when women are age 30 or 35, and then determine high, middle, and low risk. And based on that information, point people to the normal screening procedure, start at age 50 here in the Netherlands and a few other countries. But if you're in the high risk category, based on your polygenic risk score, maybe you should start immediately with that mammography screening program every year, for example. So that allows you to stratify citizens in this case, so healthy people, based on their genetic risk for this major cancer and deadly cancer, allowing them to be very early in detection of any tumor growth there. And that allows you to be much better for preventing the development of this tumor. For example, the current screening program in the Netherlands, which starts at age 50, still misses 20% of the breast cancers. So that proportion can be maybe detected using this polygenic risk scores for a large part. So this holds for breast cancer, where we have screening program, but also might be true for prostate cancer, colon cancer, lung cancer, where screening programs are either underway in several countries or are being discussed by other countries. So this is again a sort of overview of where we see applications of using genetic information, so not only PRSs, but also genetic information in a broader sense. For example, pharmacogenetics and clinically actionable variants like FH mutations. Those can be used in clinical settings, either here in hospitals, academic hospitals, or maybe by general practitioners or pharmacies to provide such genetic information to patients, but also to, of course, to the clinicians to help better understand the situation of that patient. And once doing that, you might also provide, let's say, some goodies like blood group typing or HLA typing or ancestry, for example, which can also be clinically very important, of course, for the treatment you have in mind for that person. We already discussed with you the national screening programs. I think there's a very good prospect for implementing the polygenic risk score for breast cancer based on the clinical trials ongoing now. But several other diseases like diabetes, obesity, and the locomotor diseases can also be subjected to that. But of course, needs a little bit more discussion. I will not go into applications outside of clinical science. You probably are all familiar with, for example, well-known examples like the Golden State Killer, which was identified eventually through the availability of all this genetic information in large databases. So there are many other possible applications, but that might be subject for another webinar here. Okay, the final part will be dedicated to the Genome of Europe, because an important part based on all these fantastic applications of genetic information was, how much genetic data is there and do we need more data actually? Well, to start with, how much is there? Well, this is showing based on personal investigations I did, also talking to companies. And what we know now that, what I said earlier, about 6 million people have been whole genome sequenced, but they're mostly in the US and the UK. And as you understand, in this current geopolitical era, that causes some concerns with people here in Europe, but also in the US and the UK, how to access the data and collaborate, etc. Well, I'll come back to that at the end. But the arrays are much more abundant because over 1 million, probably 150 million people by now have been genotyped by using arrays. But again, the news is there. This is mostly in the hands of commercial companies like 23andMe, Ancestry.com, MyHeritage, etc. And these are also mostly US, UK based. So that causes some issues. And if we look in the situation in, let's say, based on a country level, what you see here is that most of the data indeed is in either the UK with their fantastic genomic programs, like our Future Health, UK Biobank, and Genomics England. Of course, US with these major programs, Million Veterans Program, the All of Us program. And there are some other countries like Canada, sequencing a lot, Qatar, the Emirates, etc. Iceland, of course, a well-known example, but a small country in Europe. But as you see, in general, Europe is lagging behind here. So that has led to some discussion in Europe. Should we get our act together and get some sequence data organized here? Well, first of all, we have to question, is that really necessary? Do we need such data? How much data is actually needed? Well, we know that from, for example, the PRSs we developed for height, that if you want to understand the height architecture, you need at least 5 million people. Now, height is one of the easier examples because the data is available also, but the architecture looks pretty straightforward here. If we look at diseases where the heritability is lower, but there's much more gene-environment interaction, take, for example, diabetes or obesitas, we might need tens of millions of people, maybe 50 or 100 million people with that genotype data. And in order to fill those genotype arrays also with human diversity, because then we're speaking not only about white Caucasian people, but about many more different ancestries, we really need to genotype many, many more people, and we really need to sequence many more people to get a good eye on what kind of genetic diversity is there, not only in the European population, but also, of course, in other ancestries. So yes, there's a big need of more genetic data. And that is summarized here. So for height, we know 5 million, but for other diseases, many more million people need to be genotyped. And that has led to the 1 Million Genomes project, and that is based on this observation that if you look at what we call the genetic density per country, how is that organized? Well, you see there are a few countries in yellow and green which are doing quite well. The Scandinavian countries mostly, and Estonia and Iceland, of course. But the vast majority of Europe is lagging behind. There's very little genetic data actually covering the genetic diversity in these countries. That was also noted by a very well-known report written by Mario Draghi, the banker who saved the euro, you might remember, 2008. And he wrote an advice to the European Commission on how to become more independent and more sustainable towards the future by investing in several strategic areas. So not only the banking system or building high-speed trains for better connecting Europe, but also investing in genomics in Europe. And he mentions the 1 million genomes 3 times, in fact, in the report. And that's why we're so hopeful that indeed the European Commission will invest, and as they are doing, as you will show you in a moment. And one example of that is the 1 Million Genomes project outlined here that started in 2018, and I joined a little bit later. It is organized according to working groups where all the experts across Europe are discussing different aspects like, what kind of data do we need? How should we make it accessible for people? What kind of IT solutions do we need to develop for that? But there are also some working groups on applications of determining, okay, for what uses are we generating those data? And one of those working groups is Genome of Europe, which I started together with my dear colleague Andrus Uitterlinden from Estonia. And as you can see here, we have a good representation of European countries, including some observer countries. As you see here, Switzerland and the UK are there for obvious reason, but also Poland has now joined the 1 Million Genomes program. And by the way, we're actively collaborating with those people in those observer countries also. Well, that has led to some projects being funded by the European Commission. You see them highlighted here. The small lines are the so-called coordination programs, the Beyond 1 Million Genome programs here on the left-hand corner. But there are 2 big projects I'd like to discuss a little bit further, in particular the Genome of Europe. because that was funded in, excuse me, in 2024 with 45 million euros in a matching format, I must say. So half of it is delivered by the Commission, but the other half has to be delivered by the countries participating in this, making those countries also invest in these infrastructures. Genome of Europe is a sister project of the Genomic Data Infrastructure project, which is developing the IT solutions for storing and making this data accessible across the European countries in a federated way. And I'll briefly discuss now the Genome of Europe project, not without telling you that we're always aware of that we are burning taxpayer money here in Europe. So we're very careful, of course, with doing the right thing with this precious money. But it also illustrates you that there is substantial investment in genomics across Europe, over 100 million now, just looking at this particular project of the 1 million genome project. Well, here the Genome of Europe in a nutshell. What we want to do is sequence 100,000 genomes, which will represent the genetic diversity across Europe. So we will try to make a representative collection, which is proportional. So bigger countries like Germany and France deliver more reference genomes to that, like smaller countries like Luxembourg or Malta or Estonia. And the Netherlands is somewhere in between there. We want to include different population subgroups in each of those countries. We asked that, so what is your population looking like? For example, in the Netherlands, we do not only have Dutch people living here, white Caucasian people by ancestry, but also people who emigrated into the Netherlands. For example, based on our rich colonial history, we have a lot of Indonesian people living here, people from the Moluccas, but also people from Suriname living here who celebrated the 75th anniversary not so long ago, but also people for more recent migrations like Turkish and Moroccan people. So we also want to include them. Oh, almost there. And we also want to, of course, collaborate with people coming from those countries, like, for example, Turkey. And last but not least, we also want to take care of the minorities living in those countries, like the Roma people and the Sami people, and want to really collaborate with them. Here you see the layout of the program. It has, of course, different work packages, working with different aspects of it. We'll not go into detail, but it's well attended. We have 27 countries contributing to this, plus the observer countries, and we have several interests from countries outside of the European. You see them listed here: Serbia, Moldavia, Ukraine, Switzerland, of course. So there's a lot of interest in this project, and that's, of course, fantastic to see, but it's a lot of work also. Big project, 52 institutes, a lot of scientists working it, coordinated by us here in Rotterdam. We had a great kickoff meeting on a boat, which is symbolic, of course, because this is a big journey we're undertaking. But there's lots of enthusiasm, and you see them all here. So the brightest minds in Europe on genomics are collected in this project, and it's really a fantastic pleasure to work with them. A few details about the Genome of Europe, which might be of interest to you. So we focus on normal citizens from those European countries and the population subgroups. It will be important for several use cases. I have one more slide, I think, on that. We are only asking for age, sex, and country of origin. So we're not asking for all these genotypes, but we ask all those countries contributing to choose their samples wisely. So coming from biobanks and cohorts which have a lot of phenotypes, that somewhere down the line we can connect it to these phenotypes. We are technology agnostic, important for today. So we are working with Illumina data, MGI data, long read data from ONT and PacBio, and maybe other technologies. We go for clinical grade, and we have many sequencing centers actually across those countries generating those data. Here you see what we have in mind as pilot use cases. So, of course, we want to recreate a much finer scale map of the genetic diversity in Europe once we have that sequence data together. But already we are looking at what you see here in the middle, use case 2, of lookups of individual clinical variants like pharmacogenetic variants or clinically actionable variants from the ACMG list. We are generating long-read sequencing data. The estimate currently is about 10% of the data will be based on long-read sequencing, but that might still grow. And of course, we also want to build a imputation reference database, and we're doing that together with Ellie Zucchini in Munich. The Dutch contribution is modest, of course, but I'd like to highlight to you it's coming as an example of how this works in practice. It comes from 3 cohort studies in the Netherlands: the Lifeline study, the Rotterdam study, and the Helios study in the Netherlands. And we're seeking about 6,000 genomes, 5,000 short read and 1,000 long read. We're very happy to have a special contribution based on our royal family. because as you might know, and you see that highlighted here, they have sequenced their own genomes, at least part of them. They didn't do that themselves, of course, but other people did that. And that's being illustrated here in a what they call now the DNA room in their royal palace, where they also receive important guests. You see them highlighted here. So ministers which are being appointed, well-known people from other countries in Europe, like Zelensky here, the Spanish king came here, and other well-known people from all different sorts and kinds. And of course, also we had a famous visitor from the US coming here, and he liked the room, and it might explain the reconstruction he's currently doing at the White House. Maybe there will be a DNA saloon there. As you know, working together comes with challenges, so that's why I show this slide of Europe, which is Europe based on prejudice, as you know. So you see the Netherlands here as Sodom and Gomorrah. This is a map, by the way, made by a Bulgarian artist, Janko Svetkov. I strongly advise you to look at his website. It's fantastic and hilarious. But it is a reality, of course. We in Rotterdam think we do things much better than them in Amsterdam, of course, and that's true also for the countries. So you have to overcome all these prejudices. So because we have so many different cultures, and you might have read the most recent safety plan from Donald Trump is also highlighting this variation across Europe and how to deal with it. It's a problem for the US, I understand now, but not so much for Europe actually. But on top of that, politics is not always helping. We have the Brexit. UK has left Europe, but now they're rethinking it again. And we have small areas in Spain, for example, who want to become independent. And then we have countries who want to join the European Union. So there are many more challenges, of course, to take into account. But I can tell you on the scientific field, we are closely collaborating. For example, here with UK, Matt Brown, director of Genomics England, good friend of mine. We met Rory Collins there of the UK Biobank and other people from the UK. I was lucky enough to meet President Obama then. He who started the All of Us program in the USA, very important for generating data in that fantastic program. We're having an active collaboration with the All of Us program with Jeff Ginsburg here. So yes, scientifically we are collaborating, but that collaboration has to be expanded further, as you can see here, because Europe is not limited to the European continent. We have a global footprint on all these other territories, and they are European territory actually, where we also have to reach out to uncover the genetic diversity on those areas. So there's much more work to do. And that's why we are developing new plans, and these are my final slides, I'll finish in a moment. We started a working group on pharmacogenetics that's led by Jesse Sven and Lily Milani. We'll not talk about it today, but for obvious reasons, that's important. And we're now discussing, okay, what's next? So we have sequenced 100,000 genomes. What can we do more? Well, the easy answer is more genomes. We need to cover much more genetic diversity, but also to create genotype data across Europe to really start implementing the use of genetic information in clinical practice and prevention under the logo of, let's say, we need less patients rather than more doctors. And that's, of course, a hard truth we have to follow. And that's why our current proposal is to sequence 1 million genomes and to genotype 50 million samples across Europe. It sounds big, it is a big project, of course, but we're negotiating this now with the European Commission. All lights are on green until now, I must say, so that's going very well. And the numbers we need, as I explained before, in order to understand the genetic architecture based on the European genetic diversity, in order to implement it also locally. And that new proposal is now currently being worked out together with, of course, many colleagues from other countries to create much more data, not only sequence data, but also array data, put it in a big database, come up with recalibrated polygenic risk scores, and design, for example, pan-European arrays, which carry all these variants to genotype all these samples. Now, of course, this will happen in close collaboration with the world. The other big data generators like China, like the US, like the UK, South America is coming up, Africa is coming up. So yes, we have to collaborate there, but those are things we can tackle, I believe. We also want to collaborate with industry. Like I showed you, we have been collaborating for the current Genome of Europe program with the big providers by negotiating not only deals, but also collaboration with them to tell them our needs and also how they can help with getting this technology implemented across those different European countries. So I very much stimulate the involvement of industrial partnerships, not only the technology providers, but also companies like Novogene and others, which can provide these services for you in case you need them in order to make much more use of genetic data. So this is my talk, but not without leaving you to say thank you to all the people in the laboratory. There are too many to mention here, but of course I stand on their shoulders. I'm just the post sign telling you this, but it's all based on their fantastic work. So a big thank you to them. And a big thank you also to the Genome of Europe collaborators, and in particular to Helen Ray Jones and Jeroen van Rooij, who are helping and guiding me through this fantastic project, and really making it a pleasure to be part of this. So with that, I'd like to thank you for your attention, and I'll be happy to give the floor back to Wailin. Thank you.
Well, thank you so much, Andre, for your very insightful and visionary, let's say, presentation, and this very great contribution and leadership to the Genome of Europe, and also globally speaking, I would say, especially combine the data with the clinical applications. And we are entering the Q&A sessions, and of course, we got quite a lot of questions, and I will pick up a few questions to start with. So the first question is that when talking about such a huge collaborative consortiums and projects, a sample might be collected differently and come with different quality. So and of course, and data will be processed by different sequencing platform method.
Yeah.
So the first question is that how can you briefly, let's say, elaborate a bit on how you standardize this procedure and to make a data and the result comes in kind of consistent way?
Yeah. Well, the short answer is we collect information and we provide some recommendations for people to start up. But we're basically preparing for heterogeneity in the data. And that's the reason is because initially, yeah, the easy solution would have been there's one laboratory doing all the sequencing for this Genome of Europe. Let's say Erasmus would do that, which I thought was a bad idea. Because that worked, of course, in England to set big projects up, but I don't see that working in Europe because of all the political situations. But also, I think more practically, in the end, this has to be implemented locally. So it's much better to set up an infrastructure in all those countries and sequencing institutes, which have to collaborate and bring those data together now. So now it's a challenge for us to work with this heterogeneous sequence data, with DNA samples that have been collected in a slightly different way, and see what kind of heterogeneity is there in order to combine all these data into a useful reference database. Because this is only the start of the sequencing, as I showed you, there will be much more sequencing data being added to this. So there will be heterogeneity anyhow. And for sure, of course, also when we start to collaborate internationally with projects like All of Us or the UK, our Future Health or Genomics England, for example. So we have to be able to cope with heterogeneity rather than try to prevent it. But we do that by providing guidelines and advice. And for example,
Okay, yeah.
Ivagat in Barcelona is doing also experiments by sending around one sample across different sequencing institutes to see how the data looks like and provide some feedback on them how to improve. Yeah.
Yeah, well, thank you so much for your elaboration. And the second question actually associated with the previous one. The question is asking about, okay, what kind of criteria are used to ensure that each country samples are truly population representative?
Yeah, that's a good one because I sometimes say I'm not the genome police. So we're of course telling people in order to make this database useful, it should be representative and inclusive. So do your best to collect as representative possible samples from your studies. Now, I showed you the example for the Netherlands. We have fantastic epidemiological cohort studies, which are very representative of the local population because of their very high response rates also. So that's an example. That's also the case in some other countries in Europe, but certainly not all. But it can also help European countries who want to set this up to say, okay, this is how we should do it. We want to set up a biobank for another country in Europe, and we should be population-based and get as many people as possible on board. And that will make sure that this particular sample will be representative for the population. But again, what we did is we asked those people, how is your population looking like? And based on that, we identified in Europe over 200 different population subgroups. Of which the money we currently receive now from the European Commission allows us to characterize 40 of them. So there are many more population subgroups still to tackle if we get more funding later on. That's why I'm negotiating for this bigger project right now. I hope
Yeah.
that answers the question.
Yeah. So during your presentation, you also mentioned the application of long read, which is third generation sequencing solutions.
Yeah.
So you mentioned that right now you have a 10% of data come from long read. And so I'm just wondering if you can briefly, let's say, comment on what kind of advantage and profit that you can gain from this third generation long read, especially
Yeah.
recently Roche also joined the game by so-called sequencing by expansion. So basically, I'm
Yeah.
asking if you have give any technical and genetic, let's say, expertise on
Mm.
the combination of short plus long
Yeah.
and of course with the microarray.
Yeah. I will leave the microarray out of the discussion because for Genomics, currently we're focusing on the sequencing, which is challenging enough, I can tell you. So we have some very bright minds on this, like Ivo Gutt and Olaf Ries from Tübingen, to help us with that, to advise, okay, the robustness of this technology and should we use it in Genome of Europe data generation. One challenge, as you mentioned, is combining short read with long read data, but we have people looking into that right now. How can we combine such data, which I think globally will be a challenge. So that's why we're also discussing this with other sequencing people, of course, around the world, how to tackle this. But it will be, of course, of big advantage to get a much better insight in structural variants, for example. As we know, short read sequencing can detect some structural variants, but is certainly not good at it. So that's why we are now thinking about for the calling of the variants, maybe we should limit that for the short read to the simple ones, so the single nucleotide and the insertion deletion, small ones, but not go for the structural variants because we have long read data that might be much better resource for that, and then combine the called
Mhm.
variants into a single file for that particular sample by having used long read and short read together.
Yeah, yeah. Okay. Yeah, I appreciate your insightful and expertise, let's say
And
comments
by the way,
on
on the
yeah.
Roche, yeah, fantastic. We're of
Yeah.
course continuously looking at this. This is a challenge for sequencing. It's moving all the time. Now we have Roche. We had Ultima before, Element. There are probably others coming in the coming years. So we have to tackle that. And again, this is a disadvantage for sequencing compared to arrays because this has been a stable technology all the time. So I think
Hmm.
arrays are much easier to work together
Hmm.
with all these different
Yeah,
resources. DNA samples
definitely.
and institutes. But yeah,
Yeah, yeah, definitely. So we talk a lot about the current genomics and the genetic progress, and of course, another part of this webinar I want to address is the future. So based on your vision, so let's think wildly. So do you
yeah,
think in the future, sooner or later, actually, people can do such, genetic analysis by themselves by using a portable device, for
Mhm.
instance, a microarray, and then transfer the data through cloud to the institute and for analysis. So basically, that's like a win-win situation, and you get more data, and the data will be processed easily, and everyone, not everyone, but people can use such a device in the future at home.
Yeah, it's technically possible, and for sure people will be doing this. And I know some people are doing this, but those are colleagues of mine who are working in sequencing lab, which have direct access to the technology. My own genome has been sequenced many times. I'm in many different databases. So people like us, let's say the more advanced, insightful people, will do this. But I'm not sure if this is really useful for the population at large, because don't underestimate the complexity of the data. And that's, I mentioned it briefly. For example, the VUSs, these are really a plague and important. Also in legal terms, because once you have detected them and they're in a database, people can start, hey, you had that data from me. Why didn't you tell me? Yeah, because we
Hmm.
learned only 3 years after that this is a clinically actionable variant. So that's why currently I would say let that data be generated and interpreted by knowledgeable people from an
Yeah.
hospital or whatever institute, and of course do that together and try to explain that to people and see how that information can be useful to them. I would not immediately, let's say, liberate the market, say let everybody sequence for themselves, because I think this will lead to chaos at the moment, and that might work against using the genetic information. So at the
Yeah.
moment, I'm much more in favor of a more structured approach for that. Yeah.
Yeah, yeah, got you. And another question is also about the future. So you are doing a great job in the Genome of Europe. So I'm just wondering if your consortium is also probably already kind of collaborating, worldwide speaking, with, for instance, Genome of Asia, Genome of America. So to basically perhaps to build a bigger foundation for more comprehensive data and to
Yeah.
basically, yeah, more diversity and population, let's say, ethnic oriented risk.
Yeah. Yeah, answer is yes. Only, of course, we've just started, and we must realize
Hmm.
our position. We're late in the game. That's for sure true. The advantage is we can learn from the mistakes made earlier. But of course, we do not have much data to offer right now. I hope that will be improved in, let's say, 2 years' time, when we have sequenced most of the 100,000 genomes and maybe more. And then we have something to offer to also these other fantastic programs in the world. I
Hmm.
mentioned a few. And that's why, for example, we have an active collaboration started already with all of us because we foresee this happening somewhere down the line. So let's start that discussion right now and see where we can learn from each other. And what I
Hmm.
said, the final two slides I showed you about this new proposal, that is much more looking like this All of Us program or our future health program in the UK for that matter, where we really want to involve phenotype data and, for example, also exposome data, which is collected in all these biobanks and cohort studies. And that's, of course, will be much more valuable to collaborate also with these other programs and do joint analysis. But I can tell
Yeah.
you there's a lot of interest, of course, in the Genome of Europe because of our colonial footprint. You know, we've been anywhere over the world, so there are always
Mhm.
European
Yeah, truly.
remnants in their genomes
Yeah.
somewhere. So that's why we are also very aware of that and are really open to collaboration. Yeah.
Hmm. Yeah, so due to time constraint there, I will just throw the last question. So I think I'm getting a new business to Andre. So one audience asks, it's possible to join the program. In our institute, we have many healthy subject already sequenced or genotype. So basically, he's wondering if he can join and how to join and who's the contact person.
Yeah. And that has to do with the, let's say, administrative organization. This is a country-driven project. So what I do if this lab is, for example, in France, then I say go to the representative of France in Genome of Europe, and they will help you to get your data on board. In principle, we also allow what we call legacy data, so that's already sequenced data, provided it is of sufficient quality and we know a little bit more about it than just having the data from a representative population sample, like we discussed before. So we want to know where are these samples coming from, what technology did you use, is it indeed deep enough? We want clinical grade type of sequence data. We're quite liberal in the starting phase now of the project, but will become, of course, more strict towards the end to have high quality data in there. But we're open to additions of other data sets for sure. Yeah.
Yeah, okay. I really appreciate your answer and expertise. And our time is running out, and I would like to round up our webinar with several take-home messages with you guys. So first, let me share my. Yeah, so as I mentioned, I want to basically round up this webinar with several take-home messages highlighted from Andre's talk. So the first point is that Europe will increase its genomic footprint by investigating in genomics programs. The Genome of Europe project will sequence more than 100,000 reference genomes, covers European genetic diversity, but of course, more data collaboration will be needed. An array are cost-effective tool to deliver actionable genetic information in healthcare and prevention. And much genetic data and many genetic analysis tools are available to implement the use of genetic information much more broadly in healthcare, prevention, and society. So thank you all for your time today, and I hope you enjoyed the webinar and found it insightful. As I mentioned, we haven't managed to answer all your questions, but we will get back to you in a later email with answers. We will email you a link to the webinar recording the slide deck, and you can expect to receive the week after. So when the webinar finished, a survey will pop up, and we appreciate if you can taking time to finish it so that we can improve our webinar and to find more webinars which is more relevant to you and your research goals. To appreciate your attendance at the webinar, Novogene is pleased to offer you a 15% discount on your project. An email will be sent to you within the next few days with a code that you can use to apply the discount. Moreover, please, there will be also a link in the follow-up email for you to view a certificate of attendance. If you need one, it can be printed from your browser windows. In the meanwhile, you can follow up on Twitters and LinkedIn to keep up to date with the future webinars, offers, and the news. We look forward to seeing you all on the next webinar. Have a pleasant day, and thanks once again, and our gratitude to Professor Dr. André Uitterlinden. Thank you so much. Have a nice day.
Thank you. It was a pleasure. Thank you.